In this video, we will talk about the types of polymerase chain reaction. So first of all, you should know what is a polymerase chain reaction. Polymerase chain reaction is a basic molecular biology technique that is used to amplify the DNA and RNA sequences. So it is basically an in vitro method that utilizes the enzyme DNA polymerase or tag polymerase is basically used for PCR. So it is known as polymerase chain reaction because it involves the enzyme DNA polymerase or tag polymerase is used in PCR. And it is a chain reaction because it involves the cycles of repeating steps, and these are repeated again and again. So that's why it is known as a chain reaction, and the products that are synthesized during one cycle are utilized in the next cycle. So that's why it is known as polymerase chain reaction. So here in this figure you can see the basic procedure or basic steps that are involved in PCR cycles. First of all, there is a double-stranded DNA. The first stage is a denaturing stage in which the DNA, double-stranded DNA is denatured to create single-stranded DNA, and it occurs at 94 to 95 degrees centigrade. After denaturation, next stage is the annealing stage, where the primers that you have added in the PCR reaction mixture anneal with the DNA strands. So annealing occurs at 50 to 56 degrees centigrade, and next is the extending stage that occurs at 72 degrees centigrade, and this is basically the extension of the DNA strand or the addition of nucleotides to the primers that results in the extension of the DNA strand. Okay, so these are the stages that are involved in a single cycle of PCR. So in these stages are repeated. Next cycle starts in which the double-stranded DNA strands that are created in the extending stage enter the next cycle, and all of these stages are then repeated. So next is reverse transcriptase PCR. Reverse transcriptase PCR is basically known as reverse transcriptase because it involves the activity of an enzyme known as reverse transcriptase. So in this type of PCR, RNA sequences are basically utilized. First of all, there is an amplification of messenger RNA strands. So after the amplification of RNA strands, we use the reverse transcriptase enzyme and primer to synthesize the complementary DNA strands. Then these complementary DNA strands are basically used for the amplification of our specific DNA strand in case of simple PCR. So basically the next steps involved in the PCR are similar to the simple PCR, but it differs from the simple PCR in that in reverse transcription PCR we utilize messenger RNA amplification, and then we synthesize complementary DNA by using these messenger RNA strands. So next is real-time polymerase chain reaction, that is also known as RT-PCR or qPCR. Real-time PCR is known as real-time PCR because in this type of PCR we observe or monitor the amplification of our targeted DNA molecule during the PCR in real time, not at its end as in the case of conventional PCR. So for this purpose, different fluorescent dyes are used that produce glow at the end of the cycle. So in this figure here you can see that two types of dyes are used: reporter dye and quencher dye. Reporter dye is basically attached to the 5' end of the probe, whereas quencher dye is attached to the 3' end of the probe. And the first stage after denaturation, the probe annealing occurs. The probe attaches to the DNA strand, and then primer annealing occurs. Primer attaches to the DNA strand. Then a primer attaches to the DNA strand. The primer, after primer annealing, the tag polymerase enzyme attaches to the primer and results in the addition of nucleotides in the 3' end of the primer. As the addition of nucleotides result in the extension of complementary strand, so when it reaches the probe, it results in the cleavage of the probe, and after cleavage, the reporter dye gets separated from the quencher dye. It produces fluorescence. So the production of fluorescence is a signal that one cycle of the real-time polymerase chain reaction has completed.